My name is Logo. I'm currently in year 3 of the Biomedical Discovery and Commercialization program here at McMaster. One thing that attracted me to this program specifically was that it is at the interface of biomedical science and commerce. So I thought I've had a very science-intensive education so far, and I thought that I wanted to gain a commerce skill set as well, and I thought this would be the perfect opportunity for me to do that. We were supposed to find an inhibitor for an enzyme called human tissue transglutaminase. And what that enzyme does is it forms links between proteins in the body, in humans as well. So the main thing was that we were trying to find an inhibitor for it because it was suspected to play a role in neurodegenerative diseases. Specifically, these are diseases where proteins clump together, either within or outside of neurons, and they eventually contribute to the death of the neurons and the person. So for very obvious reasons, we wanted to find a way to inhibit the aggregation or the clumping together of these proteins in the hopes that we could improve outcomes for people who suffer from these diseases. So we found, unfortunately for our project, we were unsuccessful in purifying the enzyme and characterizing it, but the main insight that we could get from this project was that we were learning more about how the drug discovery process contributes to the treatment of diseases. So we have a protein target. We're trying to find molecules that can stop the protein target from causing whatever harm it is causing. And so we gain a lot of insights in being able to analyze scientific data relating to all of this, including just looking at images of gels that show our protein, looking at graphs that show the different activity levels of the protein, being able to really just take data from very information-dense papers and turn it into something that you can talk about in front of a wide range of audiences. We're measured on our ability to ask questions about why things didn't work as well. So, one of the beauty about having, a really large research course, so it is 9 units, that's equivalent to 3 courses spread out over the whole year, is that we had enough time to try something. If it didn't work, ask questions about why it didn't work, investigate why it didn't work, and then figure out a way to make sure that the next time it'll work just a little bit better. And I've always been told from my graduate school friends and from researchers who've given talks that everything doesn't always work the best the first time. In fact, it doesn't. Most of the time, it doesn't work great the first time. But as long as you're able to ask those questions about why things didn't go according to plan, you can create a new plan that might succeed in the future. So that's the key. We went all the way from the beginning. We expressed the protein, meaning that we generated it in a bacterial cell line. We were able to ask questions about why expression wasn't successful the first time around. We were able to we were able to run images of gels that showed whether or not the protein was expressed, and we were able to analyze that as well. It was very presentation heavy, so we were able to take all of the information that we generated from all of our experiments and communicate it to an audience that is consisting of fellow students, researchers here at McMaster, or individuals who maybe don't have the science background but are really interested in the science still. So just being able to communicate our findings and being able to find them as well is really a huge draw for this program for me. So one thing I know now that I didn't know when we started this project was just, as far as the specific enzyme that we're studying goes, I know a lot more about that now than I did before. And just the issues that are associated with, running an experiment. There's a lot of self-directed learning in this program, especially with creating, the protocols or like creating the plan for what you're going to do when you actually get into the lab. So I learned a lot about creating a plan for entering the lab and being able to follow it even if things don't go according to plan. So just that soft skill of being able to plan something out and then implement it and then change things up when they don't exactly work, it was a huge development opportunity for me in this program.